Yeah, so what we're going to talk about today is the first lecture of the series, I'll introduce just the basics of neuroscience today and pretty much the cellular structure. And so first of all, I should introduce myself. So my name is Arnav, and I'm a newly hired assistant professor at the Center for Neuroscience. So if you go to my website, so I research on the circuits that control pain and itch behavior. I look into how the different population of neurons. So as you know, the brain is composed of billions of neurons, and they can be broken down into different circuits, just like a circuit board in a computer, and how each of the circuits they generate a very specific kind of behavior. And for me, it's behaviors that are generated by anything that's painful or can make you itch. So that's about myself, and so we'll just go on with the class and the coursework. So this picture on the left is a picture drawn by Ramani Kahal. So he was a neuroscientist. He got Nobel Prize in early 1900s, one of the first Nobel Prizes that were awarded in medicine. And he was an artist and scientist. And this cell just tells you how intricate a neuron can be. So neurons are basically the fundamental units of our brain and our nervous system, and that's what controls everything that we do in our daily lives. And when there is something wrong with it, it can cause devastating disorders. And in a way, if you think that or imagine that mental disorders are actually the worst, because at least for everything else, you know what is going on with you. But when you have a problem with your nervous system, in most cases, you don't even know what you're going through, because your perception or what you understand is controlled by that, right? So if something is, if something is wrong with the CPU, it's much harder, it's much a bigger problem than a monitor or keyboard. So that's where the importance lies of having a basic understanding of neuroscience. And this picture tells you this is one cell, and you have billions of cells. So if a cell is so complicated, and what like a billion times of this can do, right? So with that, we'll start how we understand what we know about neuroscience, the current state of affairs, and we'll go through, we'll try to cover that in 10 lectures. And I would just like to mention that, like the modern neuroscience, the cellular molecular neuroscience, it's only around 120, 130 years old. So what are we going to study is being discovered, or has been discovered over the last 100 or so years. So this is a picture of Ramani Kahal, and I stress on it because I stress on his career and his findings is because a lot of the most of this, so he was active in late 1800s, end of 1800s, and early 1900s, and a lot of his findings are still valid in at least for cellular and molecular biologists or neuroscientists like me. And he was armed with something very basic as the microscope you can see. And he would basically see tissue sections under the microscope and would draw by hand. And surprisingly, even we're armed with very fancy microscopes and high-end technologies, but our findings our findings or understanding is not very much different from what he found or what he predicted. With that, I'll just show you. So on the left is one of his drawings, which he drew.
Sir, if I might interrupt, for some of us, the slides are not changing.
Oh, really?
It's turning up for many of us.
For all of you, or?
Many of us can't even see the slides.
For some of us, the slides aren't changing.
Okay, 1 second.
Okay, now it just did, yeah.
So I did full screen, and maybe that's the problem. So is it fine now?
Yes, you're on the
Yes,
third
now,
slide, right?
now we can see it. Yes.
Yeah,
Yes,
yeah.
sir.
I can. One second. If I do that. Okay, so this is Ramani Kahal, and so yeah, so I'll just go like this. Okay, so this is fine, right?
Yes, sir. Yeah.
Okay.
Yes,
Uh,
sir.
Yeah, so as I was talking about Kohal, and so on the right is something very modern and within the last 3-4 years, and on the left, so on the left is a drawing of a Purkinje cell in the cerebellum. On the right is actual Purkinje cell in the cerebellum that has been filled with the fluorescent dye and been imaged with the microscope. So you see like how accurate he was, even though it was more than 100 or more years back, like, it was done 100 years back. So with that, so now how did he come to do this? So there was another brilliant scientist called Camillo Golgi, and he was in Italy. So what he found was he found a way to stain neurons, fix and stain neurons. So what he did was, so before him, the problem was that there were not many ways to visualize neurons. So there's only Nissl stain, and Nissl stain would only stain the cell body of the neuron. And as you saw, a neuron have very intricate processes. So it was very difficult to see that how neurons are different from other types of cells in the body. So he developed this staining technique where he fixed the tissue in potassium dichromate, and then the tissue kind of became hard, and then he dipped it into silver nitrate, and the silver chromate, these metal deposits were impregnated in the neurons, and for reasons that we still don't know why, the neurons not all neurons will take up this stain, and only very few neurons will take up this stain. So very sparsely, and it's very random. But the advantage was that we could see the neurons in isolation. So you can say, okay, like, how one neuron looks like. And but however, the problem was that there was existing theory that how neurons talk to each other. So this comes from the cell theory. So the cell theory basically says our bodies are composed of billions and billions of cells, and they are all individual entities, and they are communicating with each other. However, the German anatomist Joseph von Gerlach, he proposed that in the nervous system, it's not the case, and they're all interconnected with each other. And they formed this called syntium, or it's like a net, it's like a fishing net. So basically, all the neurons are continuous bodies, and they are talking to each other that way. However, there's another doctrine in 1800s. So this is proposed by Wilhelm Hess. He said no, neurons are individual entities too, and they're the whole system works by communicating separately with each other. And Golgi believed the Gerlach hypothesis that neurons are basically all like kind of mixed. They share the protoplasm, and they're all interconnected. Whereas Kohal believed that no, they are separate entities, and they are connected through some kind of communication, right? They didn't know that time, and they didn't have the technology to actually, because as you could see here, these neurons are separate, but they knew that they're not labeling every neuron, so they couldn't absolutely say that these are continuous or they're actually separate entities. But if you look at the picture, you would say that, okay, like these are separate cells. So however, Golgi refused to believe that, and when they accepted the Nobel Prize, basically there was a ugly fight that, but Kahal was right eventually, and he was proved right. And so the problem was that the light microscope that we're using, they has a theoretical resolution up to 100 nanometer, up to 100 nanometers, but the synaptic cleft, or the space between two neurons, is around 20 nanometers. So they couldn't actually resolve it, right? So they couldn't say they are different for sure. But only in 1950s that it was found that, so when electron microscopy was discovered, which has a resolution up to 1 nanometer, actually it was proven that Cahal was right, and each neuron is a separate property, and they have a space between it, which later was discovered as a synapse, is a chemical synapse. So electrical stimuli will come from one neuron, and it will cross as a chemical signal to another neuron, and again the electrical. So there's electrical to chemical to electrical conversion. And in recent times, there are other microscopy techniques which is called super resolution microscopy. So where also can resolve below 20 nanometers, and it was again proved that the neurons are separate entities. So I just wanted to show you guys that how they look like under in a science lab or under a microscope. So this is electron microscopy. And this is a frog neuromuscular junction. So what it is that so neuromuscular junctions are the connections between the motor neurons and the muscles, which means that everything we do, we extend our limbs, however we controlling any muscle in our body is through neuromuscular junctions. So the motor neurons are these neurons or the cells that are in our spinal cord, and they will extend the axon to the muscle. So anything you want to do, the signal is coming from your brain to the spinal cord, from spinal cord to the muscle, and that's how we are flexing or contracting our muscles. So this is a frog neuromuscular junction, and this is synaptic. So this is the neuron, end of the neuron, this side is the muscle. And these small round things are called vesicles, and this is how the neurons are talking to the muscle. So as long as enough number of vesicles are fused to the membrane, so a signal will go from neurons to the muscle, and then the muscle will contract. And if you think about it, it takes a fraction of a second. So you can imagine that how fast this communication is, even though you are seeing so many components into it, right? And this is super resolution microscopy. So it doesn't have that much resolution, that high resolution as electron microscopy, but still here the red and green, the red is presynaptic. Let's not get into the details of it, but the red is the presynaptic, the green is postsynaptic, basically showing similar things. So white fluorescence is more interesting because when we use electron microscopy, it's pretty much you can use any other way to mark the cells or the component of the cells. You have to kind of know what they are. However, in fluorescence microscopy, we know different components of cells or different types of cells. They express different kind of proteins, and we can label the proteins with different kind of markers, and that's how we can visualize them. So it gives us way more. We can study different things using the technique, which we cannot do with electron microscopy. So anyway, so that was the current state of affairs, right? And also just wanted to mention that so this is Golgi staining, and it's still done in different labs, and you can see that sparse neurons are labeled as the cortex. But there are other techniques or other fluorescent techniques that we can do the similar experiments, and here the different neurons are labeled with different colors. So you can see, but still the Golgi staining kind of stands apart if you want to understand neurons at a anatomy of a neuron at a single level. It's still sometimes still the technique to go for. So moving forward from that, what is a neuron? What are the typical parts of a neuron, or very basic definition of a neuron? So neuron is a cell. Is structurally and functionally almost unlike anything in our body. So there is a cell body which is similar to other cells, and there's a nuclei in it, but they will extend these processes. One is called a dendrite and one is called an axon. So these are called neurites. And then so basically when the cell is born, the cell will send a dendrite out, and this is getting signal from other cells. Whereas the axon out, which is giving cells to other cells. So that's how it forms connections and circuits. So, say you have a neuron for vision, right? So this is a vision neuron in your brain. It will receive the input from the eye, and it will give the input to another set of neurons. So if you say you're walking and you saw something and seeing something, you should stop, otherwise you will get hurt. So what we'll do is the eyes will send the signal to the neuron in your brain. I mean, this is a very simplified version of it, obviously, and it will give a stop signal to another group of cells, and then you stop. So, but of course, there are hundreds and thousands of neurons in that circuit, and this is how. But this is a basic diagram of how things work. So the synapses can form on the cell body, on the dendrites, the inputs to this neuron, the information where it's coming from, and it gives out information to other neurons. And so this is how. So this is of course a diagram, but it's a hand-drawn diagram from the book from Candle. But this is how it looks like in a lab experiment. So this is it's a cultured neuron, which means that you took out the neurons from a mouse embryo, and then you are growing them in the laboratory to study. But you can see how exactly similar or precisely similar it looks like to the diagram. So now understanding the cell biology of neurons. So it has, in a subcellular level, it has the similar components as a regular animal cell. So it has a nuclei, it has nuclear membrane, it has a cytosol. The cytosol is a little different in the sense it contains higher potassium. So when you later we'll get to that, like what are the ions that are required to convey action potential, and you will see that this high potassium is required in the cytosol. It has Golgi apparatus, endoplasmic reticulum, mitochondria, vacuoles, so neurons will excrete proteins, and this is something that you will also go through in cell biology. But I'll just go through shortly what all of them do. And so this is a neuron, this is from the textbook, and you can see it has very much similar components, even though the processes are much complicated, so there are differences, but the basic mechanisms are kind of similar. So, so one of the fundamental function of a nuclei in a cell is to produce the what do you call the genes or produce the messages for the protein. So so if you just go back a little bit, so all our cells has basically the same genome and the same set of genes, but how different cells are different, how cells in different organs in our body differ from each other depends on the genes they transcribe and the proteins they make and the proteins they express. That's how they look different, they work different, and they also kind of involved in different processes or physiological processes. So the fundamental difference is how and when the genes are transcribed, and this is all done in the nucleus. So you have the DNA, and then you have a transcription which will make mRNA. So then the mRNA will come out from the nucleus, and before it comes out, so there is something called splicing. So just in short, you have introns and exons. So what it means that what I mean by that is, so this is a piece of DNA, and then you have a gene which is basically which encodes for a protein, and it will have a promoter region. So the promoter region will decide when and where this gene is transcribed or expressed. Then you have exons and introns. So what it means by that is that so a bunch of exons form. So a bunch of exons together encodes the mRNA for the specific gene of interest. But all these exons are also kind of interspersed with non-coding regions. So these are the coding regions, non-coding regions. There is something called gene splicing, and these introns will go out, the exons will be sewn together and form the functional mRNA. So this is something that's you will study in other chapters of biology too, other than in cell biology, I guess, and molecular biology. But the basic thing is same. The basic phenomena is same. Sorry, 1 second. The basic phenomena in same in the neuron. Yeah, and so what happens after transcription is that there's the mRNA will now go to ribosomes, and the ribosomes are there are like two kinds of ribosomes, which we'll see here. One is a free ribosome that's floating around in the cytosol, and it said that the mRNAs are transcribed into proteins in the ribosomes, which are the free-floating ribosomes, and they stay in the cytosol. And if the protein needs to go to the membrane or get out of the or secrete out of the cell, it has to go through the ribosome that is stuck to the endoplasmic reticulum. So endoplasmic reticulum is another organelle, and which is called the RAF-ER. So the RAF-ER is basically the endoplasmic reticulum with a lot of ribosomes in it. So that's why it's dotted and it's rough. And that's what's processing the mRNA that needs to go to the membrane and secreted, whereas the ones that are bound to the mRNA that are bound to the free-floating ribosomes are staying inside. So why this is so important? Because as you will again will discuss at length in the later chapters, that the proteins that are expressed in the neuronal membranes or secreted out are extremely important for neuronal function, extremely important for development, and also maturation. So yeah, so and then so that we are talking about the rough ER or the rough endoplasmic reticulum. Then we also have smooth ER, which doesn't have they don't have the ribosomes, so that's why they're called smooth. So there are otherwise they look very similar to the rough ER, and that's said to be involved in protein folding and processing, which means that the proteins as it is made from mRNA are not sufficient to function. They has to be folded properly into a three-dimensional structure. So that structure is very important for the individual proteins to bind to other proteins and actually go through a signaling cascade. But if they're not folded properly, and a lot of diseases are because of that. And so that is important for its normal function. And the Golgi apparatus does post what is known as a post-translation modification and sorting. What I mean by that is the proteins now, they are almost ready, but they are not perfectly ready yet, and they need several kind of modification, which can be a chain of carbohydrates. And they need to also go to a different, say they either need to get out of the cell, they need to go to the axon, or they need to go to the dendrites, and that is decided by the Golgi apparatus. So this from transcription, which is starting in the nuclei, to it gets exported out of the nucleus to the cell body, it binds to the ribosome, it gets translated into proteins, then it is modified, then it's sorted, and then it can go to where it needs to be done. And everything has to go in tandem and to perfection to actually the cells to function properly. And there are diseases which can go through later that can happen because of misfolding or wrong post-translation modification or deficits in cells sorting. So, and then the most important other organelle is the mitochondria, which provides energy. And it's now the mitochondria are very similar to other cell types, but the mitochondria can be numerous depending on the region. So we now know what's so the mitochondria, as you know, it provides energy to the cell, and without mitochondria, the neurons cannot function, and neurons need a lot of energy because the neurons can send axons which are up to meters. So for example, a motor neuron, which the cell body is in the spinal cord, but it sends its axons to the tip of the finger. So if you want to control your finger, the signal is actually coming from the spinal cord. So, and this is one cell. So you can imagine that a cell goes that long just to send information, and how much energy would it require to do so. And it has to provide energy immediately. So, and the energy all this energy comes from mitochondria, and mitochondria are enriched at the synaptic, where the connection is made between muscle and neuron. It's present on the muscle side too. It's present in the cell body and the dendrites. And depending on the energy that is required, that's how the population of mitochondria is made. So the mitochondria has this structure, which is the fold circle cristae, and then you have the outer membrane and inner membrane. And it goes through this process called Krebs cycles and electronic transport chain, which you study probably in biochemistry. And where one molecule of pyruvic acid, that's this, that's what it takes in, and it comes from protein, sugar, and fat, and that comes from your diet, and that is converted to 17 ATP molecules. So that's if you read about Krebs cycle and etc., that you will learn it there. And as I said, the neurons need a lot of energy. It has a lot of mitochondria. As we know that our brain consumes the most of energy. It's a very energy hungry part of your body. So that's why that's how it's regulated. So then we'll go to the neural membrane. So the neural membrane, again, that's what protects the neurons from outside. It maintains a very constant and different environment for the whole thing to function. And also it's very specialized in the sense that the neural membrane in the soma, dendrites or axon terminals are quite different. And mostly because of the proteins they express. So the proteins has to, as you can imagine, to go to this specific protein has to go to specific parts of the neuron, again, to function properly. And we'll cover that at length in the later chapters. So then the cytoskeleton. So now the neuron, it's a, the membrane is like 5 nanometers. So how would it maintain its structure? So it needs a skeleton, right? So just like our body skeleton, it needs a skeleton to maintain its 3-dimensional structure. And how would it do that? And so it has these proteins called the filaments, or the cytoskeletal proteins. And so it's mostly composed of, now again, this kind of simplification for this purpose, but there are tubulins, there are neurofilaments, and there are microfilaments. The microtubule, neurofilaments, and microfilaments. And as you can see, they have different average diameters. So what would happen is these tubulins, they run from the cell body all along the axons, except for the terminals. So they're maintaining the axon along the length, the structure, and they have intermingled with neurofilaments and microfilaments. So, and the tubulins are mostly polymers of globular proteins, and so each join, so there is polymerization, depolymerization, all these events, and that's how these tubulins are built. And as you would see during when this, our body grows, say, like, I was giving you the example of a motor neuron innervating, say, a tip of a finger. And as you grow, your hand also grew, the size grew, and so the axon, to maintain its position, it has to grow along with your rest of your body. And there are hundreds and thousands of neurons like that. So how would it grow like that? And so it goes through this process of polymerization, depolymerization to grow and to function and to renew. So a neuron, unlike many of the cells in the body, they are born once, they're born when you're born, and they die when you die. So it has to maintain this structure for a long time, so it regenerates itself. So that's why it goes through its polymerization and depolymerization. And also, it has other functions. So as you can imagine that, as I said, that it can have very long axons and really small dendrites, but the nucleus is one right at the center. Most of the protein synthesis happens in the cell body, or all of it. And the protein has to now go down the axons. And how would it go down? So this is a example, a couple of experiments it's mentioned in the textbook. I got quite fun, that what is called Wallerian degeneration. So if you cut off the axons from cell body, the axons die. So that means that the axons require the food, or the fuel, to say, or the proteins to survive from the cell body. So, and this is named after the discoverer, and it's called Wallerian degeneration. And also another experiment was found by Paul Weiss, is that when he tied up the connection between axon and cell body. He saw that one side of the cell body, or one side of the tight part has concentrated protein. So it means the protein needs to flow down. But what he saw that, when he released it, that it's moving way too slow. And if it goes that slow, then it would take forever for it. But if you go through our, say, from the spinal cord to the tip of the finger, so you need ways for the proteins to transport. And that's where these tubulins and the proteins, the tubulins come into play, which is basically acts as railway tracks, and they are proteins that would take it forward and also proteins that would take it backward for the communication to happen. So therefore, for anterograde is kinesin, for retrograde transport is dyne. Yeah, and then also the different parts. So as I said, now talk about the cell body. Now we have the axon where all these microtubules are, and the protein composition of axon membrane quite different from soma. And this is because the axon potential, now it receives the communication from the presynaptic side or the input neuron. It comes down the cell body. Now it has to be transported down the axon. And there are different kinds of axon in the body. There are different purposes. Some needs to go slow, some needs to go fast. And this all depends on the speed of the axon, basically depends on the diameter, and how the speed is regulated. Again, we'll talk in the later chapters. So, and the axon terminal is quite specialized too. So the picture, if you remember, I was showing you of the electron micrograph of frog neuromuscular junction. It exactly looks like that. So the presynaptic has a lot of synaptic vesicles. So as I said, that electrical signal is turned into a chemical signal in the axon terminal, and then when the vesicles release, so all the chemical neurotransmitters are released, received by the postsynaptic side, it will become an electric signal again. So there is a conversion from electric chemical to electric, and that all happens at the synapse. And and the synapse is basically between the axon terminal and the dendrites or the cell body of the postsynaptic cell. And they also call this in person synapse, where they make the passing axons are making connections on dendrites. And so dendrites, this is how they look like. This is a three-dimensional reconstruction of a dendrite. And so what I mean by that three-dimensional reconstruction, so they take very fine sections of electron microscopy, electromicrographs, and then they join it together, and using computational tools, they will make actually how it looks like. So this is probably 1/100th of a dendrite, and then you can see the structure which is called spines, which are actually the synapses or the postsynaptic cells. So this is and also the synapse is quite specialized, and then more recently it was found that if you remember, there are no ribosomes in the axon terminals, but scientists have found that there are ribosomes in the dendrites, which means that there's a lot of local translation. So proteins are coming from cell body, as well as the dendrites are kind of dynamic that way, that they will make new proteins as required. Now, what are the types of neurons, and why is that important? So as I said, as you know, there are billions of neurons. So if we want to, and scientists often when the question is so complicated, scientists often take a reductionist approach, which means that you would like to kind of reduce the neurons into several types, in the hope that if they kind of look similar, they might function similar. If they're the same place, they might function similarly, or they might, if they express the same protein, they might function similarly, just to hopefully better understand it. So one of the more primitive ones are unipolar or bipolar or multipolar, where they have two neurites, just one neurite, two neurites, or multiple neurites. or calling them or naming them or classifying them by shapes. So in the cortex, there is a type called stellate cell, which looks like a star, or you have pyramidal cells, which has this kind of structure. Also, they can more modern or more current way of classifying them is by gene expression. So this is a very recent paper where they have sequenced the genes that are not the genome, but the mRNA, the proteins that are genes that are expressed, which is called exome sequencing, of all the neurons in the entire nervous system. And they found there are different genes that are expressed differently in this population. And I mean, this is more of a whole nervous system thing, but now we have, we can classify each part of the brain or each part of the nervous system by markers or the genes that are expressed. So then using other techniques, you can now manipulate these cells in the sense you can just activate a small part of the brain and ask, what do the neuron do? Or what do the mice do? So if it's say like, for example, it's a feeding neuron or hunger neuron, right? So, only when you are hungry, those neurons are activated. So what if you do an experiment where you found out what these hunger neurons are using this kind of techniques, and then you activate those neurons or you drive action potential in these neurons, then the mouse will be hungry, even though say the mouse has eaten or mouse have eaten to what extent they can eat, but still the mouse will become hungry. So that's how right now, the state of affairs is that we can actually go to small population neurons, drive their activity or silence them, and ask the question, what is differently this mouse doing when we are not activating or silencing them? And then the other cell type, major cell type, is the glia. So, our brains have almost same number of neurons in glia, and neurons we understand way more than glia, and glia is like, it's just, so most of them call astrocytes, and they are thought to be just providing food for the neurons or protecting the neurons and discovering the synapses and maintaining a safe environment for the neuron to function. But more recent findings that that's not true. Astrocytes a lot more function. It sometimes it expresses the same receptors as the dendrites or the postsynaptic site, and they would modulate the activity. And without astrocytes or if the astrocytic function is defective, then we're finding a lot more neurological disorders. And also they have other functions, which is, as I said, that axons, the axons, they send information at different speeds, and that is determine the diameter of the axon, but also it is also determined by the glial cells that ensheath the axon. So the axons are completely covered mostly by these glial cells called oligodendrocytes, or the myelin, which is known as the myelin. So the myelination, the amount of myelination, the amount of glial fat contained also determines the speed of translation. All these things will get into much details in the later chapters. So as I was saying, the myelination determines the speed. So what is myelination? So along the axons, there are these cells covering, forming this myelin sheath, which is called the structure is called a myelin, but the cells that are forming, these are living cells that are forming this sheath, which is called oligodendrocytes. So, and then you will see this also gaps between two myelin sheaths, and this is called Neuro. And what we'll learn later that how the action potential can jump all through the gap and like that. So that's how the speed of transmission is determined that way. Again, we'll get into this much more details later. This is harder to explain like this. But it's important to know the structures, what is the node of Ranvier and the myelin sheath and the components of the myelin sheath with the oligodendrocytes. And so this is more classic functions, and then more recent functions. There's a system called lymphatic system, which is basically the astrocytes, not only covering the synapses I was talking about before, providing food for the neurons, they're also helping to clear up debris or metabolic waste from the nervous system, or that's produced by the neuron. So what it means by that a lot of, just like anything, a lot of wastage is produced by the neurons in the brain or the nervous system, and it needs to be cleared up. And while we sleep, this glymphatic system, which is controlled by the astrocytes, they are helping clear up all this metabolic and other waste. And there's also another cell type which is called microglia. So this is from a more recent experiment where using the genetic techniques I was talking about, they have labeled two different population of microglia in two different colors. And these are also a good population of cells in the nervous system other than astrocytes, oligodendrocytes, and neurons. And these have a different origin, but these are activated when we have an inflammation, and also helps clear up when you have dead cells and something like that. So that's basically so what I try to go through is the basic components of the nervous system and the very basic mechanisms. And in the next lectures, we'll go to much more details. Okay, so with that, so that's end of that. And questions? Yeah, Chinman.
Sir, would you be covering neuronal repair in this course?
If it's in the syllabus, I will. So I have to. I didn't plan all the chapters, but yeah, we'll get into a bit definitely.
Okay, sir. Thank you.
Yeah, the next.
So with respect to slide 8, you had mentioned some slide 8. Yeah, the coloring. What is the basis for this coloring?
Yeah, so these are dyes called dyads. So these are taken up by neurons specifically. We don't know exactly why they take it up like that, but if you inject a mix of colors, then they are basically randomly taken up by different neurons. So hoping that so I'll go back to a little more basic. So neurons in any part of the body, they're sending axons to different parts, right? So if you inject the dye, depending on where they are terminating, you will get the cells marked differently. Does it make sense?
Yeah. And slide number 20, you had mentioned something about back signaling.
Yeah, retrograde.
So what exactly is that? I didn't get it.
Yeah, so the proteins, they need to go forward as well as backward, which means from axon to the axon terminals, and from axon terminals from cell bodies to axon terminals, and from terminals to cell bodies. And so when it's going from the cell body to the terminals, it's called the anterograde transport. And if it's going backwards, and it's called retrograde transport.
Okay.
Yeah, that's the nomenclature of it, right? So, yeah, Rashi. Yeah, go ahead.
Right. So I'd initially raised my hand, but I had the same question as Rashi about the differential coloring of the neurons. So it's solely based just on where the axons end?
Yes.
And
So
it
the
has
color,
nothing to do with the population subtype.
no. These are just dyes. So these are not like genetically, these are not like so nowadays you would do that by, depending on the marker the population neurons are expressing. But this is just using chemical dyes, so it's not that.
Okay, thank you, sir.
Yeah. Anybody else?
So yeah, can you explain both these experiments one more?
Which one?
The ones in this slide.
Oh, so the watering degeneration is basically like when if you give all the survival factors, given all the survival factors are there, if you separate the axons from the cell bodies, the cell bodies survive, but the axons degenerate. So the interpretation being that the axons have all the mechanisms to make proteins that is required for its survival, a solid cell body, but axons don't have it. So axons get it from the cell body. So it needs to be connected. So that means that the proteins that are required for survival needs to come from cell body, and the question then becomes how does it come down, right? That is one. And the other thing, the Paul-Weiss experiment is they basically blocked the transport and then released it, and they saw that the proteins that are coming down are coming down really slowly. So that's just by diffusion, and that is way too slow for a neuron to carry on its usual function, which is really fast. So there needs to be a mechanism for the fast axoplasmic transport, and that's done through this protein called kinesin and dyneins. Does it make sense?
Yes, sir. Thank you.
Yeah. Yeah. Anybody else?
I just had a question about the recommended rating. What's the recommended rating for the course?
So I can so for now the textbook chapter, and then so this is my very first class at ISC or like ever, but I'll upload that by today, right? So the book chapter and plus I'll couple of papers I'll upload.
You've mentioned the textbook. So what is the textbook?
Oh, the textbook, you guys don't have the details for it, right? So 1 minute. So I have the PDF of it. If you, I can upload that too in the in this group.
Yeah.
So this is a chapter 2 basically of the. So it's the 1 minute. Let me go. Is the Bear Connor? So I somehow I thought you all have the information about the textbook, but let me check. Yeah, it's the Mark Baer, Barry Connor, Michael Paradis on neuroscience, exploring the brain. But I'll upload the book chapter too. So we'll follow the book chapter all through the 10 lectures. Hey, sorry, we'll follow this book for all through the 10 lectures.
Would you also be uploading your presentations?
Yeah, sure. I'll do that too.
Yeah. Thank you.
On which days will we have the lectures?
What's that?
On which days will you have the lectures? Is it Tuesday and Thursday?
I think Vinod scheduled it. I think, yeah, Tuesday, Wednesday, and Thursday.
Okay, so 3 days a week?
Yeah, so I think Vinod, can Vinod say when did he? I can't.
Excuse me.
Yeah.
So I think Wednesday is a TA class. Tuesday and Thursday is a regular class. So TA class now. So are you taking the class on this Wednesday?
So I can okay. Then I can take the. So I'll upload the class materials and I'll take Tuesday and Thursdays.
Yeah, Tuesday. So they can do some clarification or something if they need some. So you can make use of the Wednesday sometimes.
Yeah.
If the syllabus is not covered, or you can use.
Yeah, sure, we can do that. So okay, then we'll do Tuesdays and Thursdays. Yeah, tell me.
With respect to like the grading for this particular module, will be the end semester, right?
Right now I have no idea, so I have to talk to I think Jane about that. Yeah, he would be able to better person to talk about it.
Yeah. So the end semester is only for this neurobiology.
Okay.
Okay, thank you.
You're welcome. Yeah. Anybody else? So all right then. So that's it. So I'll upload the book, the PDF of the book, and a couple of other materials. All right. Okay. Bye. Thank you.
Thanks.
Yes, sir.